Good evening to all of you. I am Dr. Shival Srivastav. I am additional professor in the Department of Physiology at All India Institute of Medical Sciences, Jodhpur, and I welcome all of you on behalf of the Education Committee, the Physiological Society of India. Today's session is titled A Review of Cardiovascular Physiology, and in today's session, we want to give you an overview of important concepts in cardiovascular physiology. As you all may remember, multiple eminent speakers from different medical institutions in India have covered various aspects of cardiovascular physiology through these lecture series. In today's session, we want to provide you an overview of these important topics. I am very grateful to the speakers who have come together and devoted their time to prepare a key summary of these topics, and in today's session we will give you the speaker's perspective of vital concepts that are essential for your understanding of these topics. So let us go ahead regarding what we will cover today. We will be talking about the electrical activity of the heart. Then electrical activity of heart translates into mechanical contraction or cardiac cycle. This will be followed by discussion of cardiac output. Thereafter, we will discuss regulation of blood pressure and then summarize the lecture with miscellaneous circulation such as cerebral, hepatic and renal circulation. So talking about the first topic, electrical activity of the heart. This topic was covered by Dr. Chitturi Vinay from AIIMS Rajkot, and Dr. Vinay in his lecture talked about the conduction system of the heart and the physiological events that are leading to the impulse generation and conduction down this pathway. He talked about the electrical conducting system of the heart wherein the SA node generates the impulse. This impulse travels down to the AV node and then to the left and right bundles and further onwards through the Purkinje fibers to the cardiac musculature. In his lecture, he covered about how impulse is generated in the SA node, what is its ionic basis, and what are the different channels that are involved. He also talked a bit about the applied aspects of knowing these ion channels and how this can be translated into clinical utility. So now let us hear it from Dr. Vinay regarding what he has to say for the important concepts of this lecture.
Hello and welcome to a quick primer on the electrophysiology of cardiac muscle. So, cardiac myocyte is an excitable tissue and thereby are characterized by specific types of action potentials. So, we have action potentials arising from the ventricular myocyte and action potentials arising from the pacemaker tissue or nodal tissue. So if you see the ventricular myocyte, it has a resting membrane potential of minus 90 millivolts. Then the entry of depolarizing current would cause a rapid upstroke through the opening of sodium channels and subsequently calcium channels in the later phase of phase 0. Then phase 1 starts with the closure of the sodium channels and the opening of a group of potassium channels called the potassium transient outward channels. This is followed by a phase called the plateau phase, in which there is continued sustained calcium entry, which is balanced out by an outward potassium current through different types of potassium channels. So this is a plateau phase because there is almost net minimal current flow out of the cell. So there is sustained calcium entry. This ensures two things: there is adequacy of calcium for contraction of the cardiac muscle, and the prolonged depolarization phase ensures a long refractory period which prevents cardiac muscle from being tetanized. Now, once this calcium channels close, you see an exit of potassium and the cardiac muscle repolarizes back to the resting membrane potentials. Now, pacemaker cells are characterized by a very unique property called automaticity. So these cells have unstable resting membrane potentials. They exhibit a property called diastolic depolarization, where at the end of the previous cycle, once the nodal cell is hyperpolarized, there is a slow increase in resting membrane potential, or this is called diastolic depolarization or prepotentials. So this is because of certain spontaneous currents. Multiple mechanisms have been postulated to explain pacemaker currents. There's a group of channels called funny channels, also called HCN, hyperpolarization hyperpolarization activated cyclic nucleotide gated channels, and then there's some calcium clocks based on the sarcoplasmic reticulum of the heart. Now, once we reach a certain threshold for opening of calcium channels of the L type, there is an upstroke. So this upstroke is slower compared to the upstroke of the ventricular myocyte because there are no sodium channels present here. There is a calcium current causing the upstroke, and eventually this would cause the opening of voltage gated potassium channels, which would bring the cell to hyperpolarization, and this hyperpolarization resets the cycle for the automaticity of the pacemakers. So one key property of both of these action potentials is ventricular myocytes have a prolonged plateau phase, and pacemaker cells have an automatic pacemaker phase 4 phase. Cardiac muscle is a functional syncytium, so there's electrical connectivity through gap junctions at the intercalated discs between all cardiac muscles. So there's an atrial syncytium and a ventricular syncytium with a fibrous skeleton between. The AV node and the bundle of his represents the only electrical connection between atria and ventricles. So as the excitation spreads across the heart, each specific cardiac tissue produces the morphology of action potential as determined by the ion channels present with it. So if you'll see, the fastest pacemaker is SA node. So you see a pacemaker potential. As it spreads to the atria produce potentials. Then there's an AV nodal gap due to slow conduction between across the AV node. This ensures that this is a gap of around 90 milliseconds between the atrial myocyte and the onset of ventricular septal depolarization. So this ensures atria contribute before ventricles. Now then there's a bundle of His which divides into the left bundle branch and the right bundle branch, and as the bundle branches propagate to the myocardial tissue, they end in terminal Purkinje fibers. This is a very rapid conduction system which ensures that there's rapid depolarization of the entire wall of the ventricle. If you see, the cardiac conduction system ensures a rapid and more importantly a sequential activation of the heart. So if you see the timelines of activation, it takes around 30 milliseconds for the electrical impulse to propagate from SNA to the AV node. Simultaneously, it's spreading across the ventricle, across the atrium. There's an AV nodal delay of around 90 milliseconds to cross the AV node. So the ventricle gets depolarized around 90 milliseconds after the impulse reaches the AV node. So this ensures atria and ventricular activated sequentially one after the other, ensuring they contract one after the other. Then there's a rapid conduction through the ventricle through the Purkinje fiber bundle of His system. Now, one very important concept to keep in mind in arrhythmias is only a supraventricular impulse can enter the bundle of His and thereby rapidly activate the ventricle in a rapid sequence, which gives us the normal ECG QRS complex. So we call this supraventricular rhythms. The normal SA node is a supraventricular rhythm. So one consequence of this sequence of cardiac excitation tissue is at any given point of time, we may have scenarios where one part of the heart is depolarized, whereas the rest of the heart is either depolarized or at rest. So there's an electrical difference between the excited part, depolarized part of the heart and resting part of the heart. So this gives us to a configuration called a cardiac dipole. And these cardiac dipoles do produce cardiac electric fields, and an ECG represents a technique to study the cardiac electric field by measuring potential difference by placing electrodes at different locations in the cardiac electric field. Right? So the ECG represents the cardiac excitation sequence. So if you see, the first part of the myocardium of the heart to get excited is the atria, right? SA node is not of enough mass to represent on a surface ECG, so that's why the first part of the excitation sequence we see is the atrial depolarization. So atrial depolarization gives rise to the P wave on ECG. Then there's a gap of the PR segment which represents the conduction across the small nodal tissue and the AV nodal conduction delay. And eventually, once you hit the ventricular septum, the first part of the ventricle to depolarize, you get the ventricular action potentials. So the QRS complex represents the spread of conduction across the ventricle. Then once the entire ventricle is fully depolarized, you get a flat ST segment. And now once the ventricular reposition sequence starts, we get a T wave. So the ECG represents a sequence of cardiac excitation.
The next lecture in the series was cardiac cycle. This lecture was taken by Professor Prasunpriya Nayak at AIIMS Jodhpur, and in the lecture, Sir talked about different events of the cardiac cycle. We all know that the cardiac cycle starts from atrial contraction, goes into ventricular contraction, which consists of isovolumetric, rapid ejection, and slow ejection phases, and thereafter goes into the cycle again, culminating in ventricular filling and then atrial contraction again. In this lecture, Dr. Nayak talked about the important events of the cardiac cycle, what were their relative durations and the factors that could affect them. So let us hear it from Dr. Prasunpriya Nayak sir regarding what is the summary of this important lecture.
Referring to the lecture 27 of the series, it is the cardiac cycle, the sequence of events that happens in your heart during one complete beat. Think of it as a rhythm of dance between the atria and ventricles, carefully timed to keep blood flowing smoothly. The time between cycles depends on the heart rate. On average, the resting heart beats at a rate of 75 times per minute, giving zero point 0.8 second for each cycle. This zero point 0.8 second is available for atrial as well as ventricles. First, we begin with the atrial systole, which is having about 0.1 second, and the rest 0.7 second is available as atrial diastole. Then comes the ventricular systole and diastole. Ventricular systole is about 0.3 second and ventricular diastole is about 0.5 second. First part of the ventricular systole is the isovolumic contraction phase. During this phase, volume of the ventricles are fixed. The ventricles behave like a closed chamber. With increased contraction, there will be an sharp rise of pressure. Next come the rapid ejection phase. During this phase, valves open, particularly the semilunar valves open, and blood rushed out into the aorta and pulmonary trunk. This is rapid ejection phase 0.1 second roughly, and then the slow ejection phase, during which the ventricular pressure is higher than the pulmonary trunk or the aorta. As a result, slowly blood pumped out into those tubes. Next comes the ventricular diastole, where it starts with 0.04 second of protodiastole, which is part of systole, in fact, which is during this phase, blood rushed out because of the inertia of moving blood while the ventricular muscles are relaxing. So this is a peculiar phase, very interesting phase of protodiastole, followed by isovolumic relaxation phase, which is around 0.06 second, and then during this phase, like that of isovolumetric contraction phase, during this phase the ventricles behave like a closed chamber and the pressure drops here. Because of this pressure drops, the atrioventricular valves open and the accumulated blood, which is during the during this ventricular systole, whatever blood accumulated within the atria, that rushed inside the ventricle, and there will be a 0.1 second of fast rapid filling phase, followed by a reduced filling phase up to a 0.2 second, whatever blood freely moving inside the atria that comes in the ventricles, it's a reduced filling phase. And then there will be last rapid filling phase of 0.1 second, as you can see from the here, yeah, as you can see here, it is matching with the atrial systole. As a result, the atrial systole is pumping, topping up the ventricular content so that it can maximize its load before it goes into the heavy lifting of ventricular systole. So that's the whole process is going on with through one cycle to another cycle. During this cycle, there are, as you can see, the phonocardiogram recording is shown here. Phonocardiogram showing the first heart sound, second heart sound, third heart sound, and the fourth heart sound. First heart sound is during the isovolumetric contraction phase, and the second heart sound is during the isovolumetric relaxation phase. It indicates initiation of the ventricular systole. This indicates initiation of the ventricular diastole. So as a result, as you can see, the clinically these heart sounds are very important. This graph is from the is named as Uyghur's diagram. Here we are showing the aortic pressure changes, left ventricular pressure changes, right ventricular pressure changes, left and right ventricular volume changes, left atrial pressure change, and the right atrial pressure change, which is all synchronized by the electrical activity of heart as shown in the electrocardiogram. Next, very important aspect of the cardiac cycle is the pressure volume loop. During the particularly in left ventricle, we are considering here, it can also be there in the right ventricle. During the isovolumetric relaxation, during the relaxation phase, during ventricular diastole, as you can see, the blood is being filled in. Where volume increases, because of the compliance, there is no much change in the ventricular pressure. Then we have the isovolumetric contraction, where pressure rises but with a same volume. So isovolumetric contraction raising the pressure, opening of the semilunar valves, and ejection. This is the protodiastole phase. Blood volume comes out as well as pressure comes out, and then isovolumetric relaxation, and it is making a loop of pressure and volume changes, which is clinically very important.
Moving over to the next lecture in the series titled Cardiac Output. This lecture was taken by Dr. Mahesh Arjundan Gadhvi from AIIMS Jodhpur, and in his lecture Dr. Gadhvi talked about how contractile activity of the heart, the cardiac cycle, translates into the physical flow of blood known as cardiac output. In his lecture, Dr. Gadhvi talked about the fundamentals of cardiac output, what are the different factors that can affect it, and what is the translational value of knowing these concepts. So let us hear from Dr. Gadhvi regarding what are the important concepts which are must know for you from this lecture.
Hello everyone, myself Dr. Mahesh Arjundan Gadhvi. I took a lecture on cardiac output, and today I am going to do a small revision of all the things which we so cardiac output, it is amount of the blood which is ejected by each ventricle in a minute, and we know that in the stroke volume, which is amount of the blood which is ejected by ventricle in each cardiac cycle, and in a minute there are how much cardiac cycles are that? It is around our heart rate. So the cardiac output, which is if we multiply stroke volume by heart rate, we can get cardiac output, and it is around 5 liters per minute. If we take body surface area into consideration, we can get cardiac index, and cardiac index it's around 3.2 liters per minute per meter square. Now we will see what are the factors which how to measure the cardiac output. For measurement of cardiac output, we can do it by Doppler, which can be combined with echocardiography, direct fixed method, and indicator dilution method. Echocardiography, which is ultrasonic evaluation of cardiac function, and it is a non-invasive technique. It doesn't involve any injection or catheter, which is there in fixed method or indicator dilution method. In echocardiography, there is a transducer which sends signals and which also acts as a receiver, and by that we can see different structures of the heart, and from that we can derive cardiac output. The other thing is direct fixed method, and before that you must know what is Fick's principle. So Fick's principle says that the amount of the substance which is taken up by any organ per unit of time is equal to the arterial level of the substance minus venous levels of the substance times the blood flow. So if we take oxygen for recording of cardiac output, so the oxygen which is absorbed per minute by the lungs is equal to arteriovenous difference of oxygen into cardiac output, and if we put different numbers into this equation, we can derive cardiac output. The other method is indicator or dye dilution method, in which different dyes are injected into arteries and the concentration are taken at different time frame, and if we put all the values, we can derive cardiac output. So these are the three methods by which we can derive cardiac output, and we can measure the cardiac output. Now we will see what are the factors which are affecting cardiac output. So for the factors which are affecting stroke volume and the factors which are affecting heart rate, both can affect cardiac output. We will see each factors one by one. So for the stroke volume is affected by preload, afterload, and myocardial contractility. What is the preload? The preload in the context of heart is our end diastolic volume. The more amount of the volume will be there in heart, more preload will be there and more cardiac output will be there. So preload, which is directly proportional to the cardiac output, if the preload increases, cardiac output increases. We discussed about preload in a lecture, and today we will see what are the factors which can change preload. So how the end diastolic volume changes, more the venous return, if the more blood is coming from the periphery to the heart, the more venous return will be there, and because of that preload can increase. Now we will see what are the factors which are affecting venous return. So there are different factors. The skeletal muscle pump, where constriction of our peripheral muscles and particularly calf muscles, which constrict the vessels inside them, and more blood will come to the heart if the muscle is contracting. Respiratory pump, during inspiration, our heart, the negative pressure inside the lungs increases, more negativity will be there, and because of that pooling of the blood occurs towards the lung, and venous return increases. Like respiratory pump, during inspiration, the diaphragm goes down, and because of that the abdominal pressure increases, more blood will come towards the heart. Blood volume, as the more blood volume increases, cardiac output increases, because it's a blood volume which is pumped by the heart. So total blood volume has directly proportional to the preload. Sympathetic activity can also affect preload. The veins, which are capacitance vessels, which can accommodate or which is stretchable. So if the venoconstriction occurs, more blood will go towards the heart. So this was about venous return. The preload can also be increased by atrial systole, because we know that 20% of the filling of ventricle occurs at the time of atrial contraction, and so effective atrial contraction or effective atrial systole can increase preload. Like that, ventricular compliance, if the stretchability of the ventricles decreases in some conditions like pericardial effusion or cardiac tamponade, in that also preload is changed. What is afterload? So amount of the resistance against which heart has to pump, it's our total peripheral resistance, and it acts as an afterload in heart. So when the peripheral resistance increases, when the vessel diameter is changed or blood viscosity is changed, so at that time afterload is also changed. Afterload is inversely proportional to the cardiac output. And the last part is myocardial contractility, the how effectively muscles can contract. So there are different factors which can stimulate. Here you can see there are different drugs or factors which can stimulate cardiac contractility or increase cardiac contractility. And there are some substance like different drugs or hypoxia, hypercapnia, or acidosis which decreases the cardiac contractility. So these were the factors which were affecting stroke volume. And there are also factors which are affecting heart rate. There are many of the factors, but here in this image you can see that if the heart rate increases, it's not always the cardiac output which is increasing. Increasing heart rate can be accommodated by changing the stroke volume. So it's not always that the heart rate increase causes cardiac output increase. So this was all about our revision part of cardiac output. Thank you very much.
After we have discussed the electrical activity of the heart, the cardiac cycle resulting from this electrical activity, and the physical flow of blood or the ejection of blood resulting from this mechanical activity, the cardiac output, now we should talk about the regulation of this important phenomena. So in this series, one of the lectures was taken by Dr. Gaurav Sharma from AIIMS Rajkot. In his lecture, Dr. Gaurav provided you with an overview of blood pressure, what are the different factors which are affecting blood pressure, and how blood pressure is regulated in its entirety by different physiological systems. This lecture is very important from a clinical and translational perspective, and therefore now let us hear from Dr. Sharma regarding what he has to say about the fundamentals or the important concepts underlying this topic.
Good afternoon, everyone. Today, we will be talking about physiology of regulation of blood pressure. I am Dr. Gaurav Sharma, Associate Professor in the Department of Physiology, AIIMS Rajkot. We have already taken a lecture about the detailed regulation of blood pressure in our series of lectures held previously. However, today we are here to discuss a brief summary or the review of regulation of blood pressure, what are the key points to be remembered and what are the key concepts to be remembered. The focus of this today's lecture will be for the post-graduations students who are looking forward for their upcoming examinations. So, in the previous lecture, we have discussed the regulation of blood pressure under the four major subheadings. Number one, we discussed about the fundamentals of blood pressure, what is the definition of blood pressure, how it is classified, and what is the clinical significance of measuring correctly the blood pressure. Then we moved on to what are the determinants of blood pressure, how cardiac output, peripheral resistance, and the blood volume interplay and regulate the outcome as blood pressure. So this is an interplay between the cardiac output, stroke volume, heart rate, total peripheral resistance, giving the final blood pressure recordings and how are they regulated. Then we moved on to the immediate control of blood pressure, where we discussed about baroreceptors, chemoreceptors, and what is CNS ischemic response. And then the last part of the lecture was what is the intermediate and long-term control of blood pressure. Today we will not go in depth as we did in our previous lectures. This will be a short video giving you the key concepts about the regulation of blood pressure. Moving on, the fundamentals of blood pressure. The fundamentals of blood pressure means definition of blood pressure. The blood pressure is the lateral pressure exerted on the walls of arteries, which is called blood pressure. We have discussed it is classified as to systolic and diastolic. Then we have also discussed what is the importance of blood pressure. The key point I would like to highlight here is that the blood pressure, when it is called is elevated, is more than 120 and more than 180 diastolic. However, in the classification, the point to note is that when the blood pressure is elevated but not in stage 1, it can be dealt with lifestyle modification, and in the stage 1 also the therapy or drug plus lifestyle can be useful. However, it is not suggested that you give only the lifestyle intervention for a stage 2 hypertension or hypertensive crisis, where we should be more focused upon giving the immediate management by drugs. And this is what is a key factor while deciding what is to be done when you see someone with the increased blood pressure. Moving on to the next part. Next part was determinants of blood pressure. As we discussed, the blood pressure is the product of cardiac output and total peripheral resistance. Any change in the BP is the change in either one or both of these factors. Now, cardiac output is itself a product of stroke volume and heart rate, and it is typically the amount of blood which is pumped per minute by the heart. In a resting state, in a normal healthy individual, the cardiac output is around 5 liters per minute. And this stroke volume itself depends upon many other factors like what is the preload, how is the myocardial contractility, what is the venous return coming to the heart, and all these factors will determine what will be the stroke volume, and that stroke volume with heart rate will determine what is the cardiac output. The second factor which is important in determination of blood pressure is the peripheral resistance. The peripheral resistance is the blood flow through a systemic circulation and is determined by the arteriolar diameter. The thing is that smaller the diameter, more is the resistance. And it also depends upon the ability of these vessels to constrict or dilate during the period so that the peripheral resistance can be altered. The third important factor is the total amount of volume of blood that is circulating. If there is a blood loss or if you are giving someone fluids, the total volume increases, which also changes the blood pressure of an individual. So these are the factors which affect the blood pressure. Then we moved on to the next, which was the immediate neural control. And immediate neural control is of three types: the baroreceptors, the chemoreceptors, and the CNS ischemic response. Now the baroreceptors are the mechanoreceptors, which are actually the receptors which respond to stretch. So when there is more or increased blood pressure, there is stretch on these receptors. These receptors are located in the carotid sinus and aortic sinus, and these detect the stretch of these receptors. Now, once the stretch is detected, these baroreceptors fire, sending signals to the central nervous system via cranial nerve 9, that is glossopharyngeal, and cranial nerve 10, that is vagus, to the medulla. In the medulla, the central processing takes place, and the nucleus of tractus solitarius integrates afferent signals, inhibiting the vasoconstrictor area and stimulating the vasodilator area. After that, there is an autonomic adjustment. Autonomic adjustment, by which we means that there is a decrease in the sympathetic outflow and increase in the vagal tone. This results in bradycardia and decreased stroke volume. Also, the blood vessels also dilate, and all these combined effect leads to decreased blood pressure when there is an increased blood pressure. To explain this concept better, we have an animation which will teach you better. So here we have a baroreceptor mechanism. We have a heart, the carotid sinuses and the aortic sinuses are marked, and these blue dots are the carotid sinuses or the baroreceptors. Now, via carotid receptors, the impulses of these increased blood pressure, that is the step receptors which activate, send the signals to the medulla via cranial nerve 9, and from the aortic sinuses they send the signals via vagus nerve or the cranial nerve 10. They go to a center in the medulla which is called nucleus of tractus solitarius, NTS, nominated in this slide. Now this sends an impulse to cardiac inhibitory center, and this center sends back the signal via same vagus nerve to the SA node and inhibits the SA node, so it decreases the heart rate. What NTS also does is it sends signal to inhibit the cardioacceleratory area, and this cardioacceleratory area send impulses in the spinal cord, which also goes and inhibits the SA node. Then what more happens is there is this nucleus of tectus solitarius inhibits the vasomotor area, and this also sends impulses to the spinal cord. The spinal cord then sends impulses causing arterial dilatation and venous dilatation. This also decreases the epinephrine release, and all this causes decreased in the blood pressure of the individual. So this is how the baroreceptor mechanism works. This is important for a lot of people looking up for upcoming examinations. The baroreceptor mechanism is a frequently asked question. Coming on to the chemoreceptor reflex in regulation of blood pressure. The chemoreceptors are located in the carotid bodies and the aortic bodies. The carotid bodies are at the bifurcation of the common carotid, and the aortic bodies are along the bifurcation of the aortic arch. Please do remember that the chemoreceptors and baroreceptors are different receptors and not the one and the same thing. The chemoreceptors respond to decreased oxygen tension or increased carbon dioxide levels or increased HN concentration or decrease in pH. Now this is how it works. When the hemostasis is decreased, then there is a disturbed decrease in oxygen, increased CO2 or decreased pH. This causes stimulation of these chemoreceptors, and which causes cardioaxillary areas to be stimulated, which increases the blood pressure and the cardiac output. It inhibits the cardiac inhibitory centers, which in turn does the same thing and increases the cardiac output and the blood pressure. And it stimulates the vasomotor centers, which causes vasoconstriction, and this vasoconstriction causes increase in the blood pressure. One more thing that the chemoreceptors does is that it causes respiratory centers to be stimulated, and these respiratory centers increase the respiratory rate, which increases the oxygen and pH and decreases the CO2 levels in the blood and the CSF. This causes restoration of the homeostasis, and hence the blood pressure, the CO2 and the O2 are retained back. These are the chemoreceptors. Now coming on to the next one, that is the CNS ischemic response. It is the last ditch effort when the blood pressure falls to less than 60 mm. There is a maximum increase or maximum effort by the body. It can increase the blood pressure to 2650 mm of Hg, and it lasts for a short duration, that is for around 10 minutes only. Now coming to the long and the intermediate and the long term control of the blood pressure. The intermediate mechanisms involve capillary fluid shift mechanism and reverse stress relaxation. The capillary fluid shift means when there is an increased BP, the fluid shifts into the capillary, from the capillaries leaks out, while then there is decreased blood pressure, the fluid is resolved from the interstitium into the blood, causing the volume to go up and down and managing the blood pressure. While the stress and reverse stress relaxation means when there is increased volume, the vessels dilate, while there is a less volume, then the vessels collapse and increase the pressure on the blood and increasing the blood pressure. For the long term effects last for days, and it does not start immediately, but it can last for long term also. It can completely restore the BP back to normal, and it functions for days and years for deranged blood pressure. Now the central point in this is the renin-angiotensin-aldosterone system, where the renin is secreted and converts the angiotensin to angiotensin 1. This angiotensin 1 is converted to angiotensin 2 by the pulmonary endothelium, and this angiotensin 2 is a potent vasoconstrictor, stimulates aldosterone, and has the various functions, like it increases the overall sympathetic activity, increases the reabsorption of sodium and potassium, causes the adrenal cortex to release more aldosterone, causes vasoconstrictions, and also increases the secretion of ADH, which helps in collecting and reabsorption of water. This whole system is called renin-angiotensin aldosterone, commonly referred to as RAS system. Now, the ADH also forms an important mechanism where there are two types of ADH receptors, the V1 receptors and the V2 receptors. V2 receptors are mainly present in the renal system, and these cause the aquaporin 2 channels for water reabsorption and increase the volume, circulating volume, increasing the blood pressure. While the V1 receptors are vascular and causes vasoconstrictions and increases the total peripheral resistance. Both these combined effect causes increase in the blood pressure. This is another important mechanism which has to be remembered about the ADH. Now, key takeaway points are the baroreceptors are the primary and rapid control, chemoreceptors are backup, and the CNS ischemic response in an emergency BP management system. For the intermediate system, we have the capillary shift mechanism and relaxation mechanism. For the long term, we have the renin-angiotensin aldosterone system, atrial natriuresis and vasodilation, and ADH system, which causes water retention by the V2 receptors and vasoconstriction by V1 receptors. And all this is integrated for the management of blood pressure, and that is the importance of teaching you all this today. Thank you so much.
After talking about the physical events leading to the ventricular ejection of blood and translating into cardiac output and blood pressure, now let us delve into the different circulation systems that are there in our body. We know that there are different specialized circulation systems such as cerebral, hepatic, renal, muscular, etc. And in this series, one of the lectures was taken by Dr. Shamreen Naaz from Vidyasagar College from Women, Kolkata, and Madam talked about different specialized circulation systems in our body, focusing on the cerebral system, the hepatic system, and the renal system. These systems have their own ways of regulating their blood flow through them, and these are important because majority of these are vital organs that subserve different physiological functions. So, a knowledge about how these systems function and how to regulate their flow and their circulation is very important. So now let us listen to Dr. Naaz regarding what are these different systems and why their circulation is important.
Hello, I am Dr. Shamreen Naaz, Assistant Professor, Department of Physiology, Vidyasagar College for Women, Kolkata, and today we will summarize the key concepts of the lecture that was delivered as part of the PSI online lecture series on dynamics of cerebral, renal and hepatic circulation. So first we spoke about the blood circulation to the brain that is divided between the arterial and the venous circulation. And for the arterial circulation, we had spoken about how the internal carotid arteries, which are paired vessels, mainly composed of the anterior cerebral artery and the middle cerebral artery, which provide blood supply to the anterior and middle portions of the brain, including the frontal lobes, parietal lobes, large parts of the temporal lobes, and deep brain structures such as the basal ganglia. These are responsible for blood flow to important structures in the brain which control movement, sensation, speech, and higher cognitive functions. We also spoke about a second set of arteries called the vertebral arteries, which arise from the subclavian arteries, and before entering the skull, they merge into and form the basilar artery. This basilar artery provides blood supply to the posterior circulation of the brain, including the brainstem, the cerebellum, and the occipital lobes. These are responsible for controlling the consciousness, balance and coordination, vision, and vital autonomic functions. We also spoke about a very important anatomical structure called the circle of Willis, and which provides the collateral blood flow, which helps maintain circulation if one major artery is blocked or narrowed. Coming to the venous drainage, we spoke about how the blood flows from the superficial deep cerebral veins into the large dural venous sinuses, and they finally emptied into the internal jugular veins. This helps to maintain the normal intracranial pressure. And we also spoke about how the blood-brain barrier, which is another important feature in the brain, which is a highly selective semi-permeable protective membrane which is formed by specialized cells such as the endothelial cells, pericytes and astrocyte end feed structures, which maintain the neurological homeostasis. These are responsible for allowing only oxygen and essential nutrients to cross the blood-brain barrier into the brain, and it blocks the toxin, pathogens and harmful molecules. We also spoke about how this cerebral circulation is very closely autoregulated by the effective perfusion pressure and the cerebral vascular resistance. These two are responsible for maintaining the autoregulation of the cerebral blood flow. Higher the cerebral vascular resistance, less is the blood flow to the brain. So we also spoke about how some chemical factors such as oxygen tension, carbon dioxide tension and hydrogen ion concentration affected the cerebral blood flow, and also the presence of sympathetic vasoconstriction fibers which lead to the constriction of the cerebral blood vessels, including reduction in the blood flow. Apart from this, we also discussed certain cerebral circulation disorders such as ischemic and hemorrhagic strokes, transient ischemic attacks, brain aneurysm and intracranial hemorrhage. So these were the concepts that we discussed in the cerebral circulation. Coming to the second set of circulation that we spoke about was the renal circulation, and we discussed here that about 20 to 25% of the cardiac output which is normally received by the kidneys is normally about 1.2 to 1.3 liters per minute, and we spoke about how the renal blood flow occurs from the abdominal aorta. It goes into the renal artery, the segmental artery, the interlobar artery, the arcuate artery, then to the interlobular artery, afferent arteriole. It enters into the glomerular capillaries, efferent arteriole, and ultimately it enters the vasa recta and peritubular capillaries, where the mixing of the blood. So the mixing of the blood takes place in the vasa recta and the peritubular capillaries, and then it leaves these arteriole and the capillaries via a system of veins which include the peritubular, interlobular, arcuate, interlobar, segmental, renal veins, and ultimately empties into the inferior vena cava. So after this, we spoke about how the regulation of renal blood flow occurs, and we mainly spoke about two responses or mechanisms which are responsible for the renal autoregulation. One is the myogenic response, which involves the stretching of the elastic walls of the afferent arteriole, and stretching of these vessels wall increases the flow of calcium ions from the extracellular fluid into the cells, and this influx of calcium ion leads to the contraction of the smooth muscles in the afferent arteriole, which ultimately causes the contraction of the afferent arteriole and decrease in the blood flow. We also spoke about the tubulo-glomerular feedback, which is autoregulatory mechanism involving the macula densa cells of the juxtaglomerular apparatus in the distal convoluted tubule, detecting high sodium chloride concentration, signaling a high glomerular filtration rate. This causes the afferent arteriole constriction to decrease the blood flow, and it lowers the GFR and ultimately reducing the renin release, thereby ensuring stable kidney function. We also spoke about the renin-angiotensin system, which is a critical regulator of the sodium balance, extracellular fluid volume and vascular resistance, and ultimately the arterial blood pressure. This regulates the blood pressure by increasing the sodium salt reabsorption, water retention, and by modulating the vascular tone of the kidneys. We also spoke about some major disorders of the renal arterial circulation, such as the renal artery stenosis, renal artery and vein thrombosis, and nephrosclerosis. Now coming to the next set of circulation, it is a very important hepatic circulation, which has a unique dual blood supply, receiving about 25% of the cardiac output, mainly almost about 800 to 1200 milliliters per minute. It involves the hepatic portal system, including the hepatic portal vein, which receives about 75% of the blood flow, and it supplies nutrient-rich deoxygenated blood from the intestines, while the hepatic artery, which receives about 25% of blood flow, it supplies oxygen-rich blood. This mixes in the liver sinusoids and are processed by the hepatocytes and leaves via the hepatic vein. So the important structures that are involved in the blood flow are the hepatic portal vein, hepatic artery, hepatic sinusoids, hepatocytes, central vein, and ultimately the hepatic veins, so which all comprise of the hepatic portal circulation. We also spoke about the regulation of hepatic circulation, especially the HABR, or the hepatic arterial buffer system response, which is responsible for the hepatic artery to act as a buffer for the portal system, which is highly dependent on the adenosine levels for vasodilation and vasoconstriction. We also spoke about the myogenic responses, the neural control, as well as the humoral factors which control the hepatic circulation. We discussed about the disorders such as the Budd-Chiari syndrome and the cirrhosis of the liver. So all these mechanisms express how we are able to understand the dynamics of the different circulations and how they are auto-regulated in our body, as well as the disorders of the circulation which happens in our body, and how our body is able to maintain these circulation. I hope you have understood, and thank you.
So this brings us to the closure of today's session. I hope all of you enjoyed listening to the perspective brought about by different medical educators from multiple medical institutions in India regarding cardiovascular physiology. I hope the lecture provided you with important must-know concepts from these respective lectures, and I hope that you enjoyed listening to this session. On behalf of the Education Committee, the Physiological Society of India, I thank all the speakers who devoted their valuable time for this revision session, and I hope that this session was useful for all the listeners. Thank you, all of you, for connecting, and I wish you all a very good evening. Thank you.